I truly would not have been able to speak. Mr. Bell threatened that I would have to sing if I couldn't, so I'm sparing you all that. I had done a literature search for a talk that I gave earlier this spring and was really pleased to find out that even though that was only 3 months ago, there had been another 6 articles published. So there's a truly growing literature on PANDAS, and a shout out to Dr. Tienemann and Dr. Frankovic and the Stanford team, because half of the new literature has come from them, and it's just a wonderful, wonderful testimony to how careful systematic research can work. They are incredibly productive, and we are so grateful that they're doing it. And I'm so excited about the surveillance study that the Canadian Pediatric Society will be doing. I think that's going to be hugely helpful data, and can't wait to get an interim report after the first 6 or 12 months and see that in the literature. And also shout out to their Calgary physicians for what they're undertaking there, as well as all the other wonderful, wonderful work that we heard about last night. It's just amazing to be here and have such a spirit of can-do. I guess that's why you're the Canadians, huh? Can-do. So in that literature, there has been a nice shift from the 2000 to 2010, where there were literally only 5 data papers and 20 editorials, most of which were speaking against the existence of PANS or PANDAS or the failings of the literature, to now where we're actually getting data on children with PANS, as well as the earlier descriptions of kids with PANDAS, including some epidemiologic studies. So I wanted to just kind of go back to our model that we talked about yesterday, and this had been the model of idiopathogenesis, a wonderful big word if you are ever scrabbling. Probably can't build this one, but it just means cause and disease mechanism, and it's all in one word, so it's nice and efficient. But when you think about that, and you think about all of the pieces to this puzzle, not only do we have sort of the cartoon bubbles of the presumed large buckets of information we need to understand, but each of those arrows actually represents processes that are quite complex. And when we talk about treatment later today, we'll be returning to this model to think about places within the model that we can intervene. But in terms of Group A strep, most of that work had been done in the 90s and 2000s on the PANDAS cohort. And as you heard yesterday, there's been a very exciting development in the basic science side. We'll talk a little bit more about this morning. Genetically susceptible host is one where I'm going to talk to you about the US consortium that we're hoping we can figure out a way that our Canadian colleagues can join us in contributing data and samples to that consortium, because we really need to get at least 1,000 children in there in order to do the kind of meaningful work that we want to do. Misdirected autoimmune response, obviously this is a huge bucket and is very, very, very complex. Not much has been done recently on molecular mimicry. You heard from Dr. Pasternak that was actually proposed a couple of decades ago, and it too has been somewhat controversial, with people debating about whether or not the immune system would be functioning that way. I think the answer has come out in favor of very clear evidence that molecular mimicry is a process at work, and so what our job is to take that information and then go back and figure out what are those molecular mimics on the strep that then are the antigens on the human host. And then finally, just what do we know about this clinical phenomenon, and what more do we need to know? And I think that every time we get an answer, it just engenders another 4 or 5 questions. So the evidence that strep was causative in PANDAS, as we heard yesterday, came from 5 lines of research. And the first of these was clinical observations and epidemiologic studies. And we didn't have any epidemiologic studies of childhood onset obsessive compulsive disorder since the 1980s. So that's a very long time ago. And things have changed during that time, particularly because of the availability of SSRIs and more effective treatments for obsessional symptoms and for anxiety disorders. We started to see that this was actually a fairly common problem. So I think if we went back into the community, we might see that it happened more often. But our colleagues in Denmark and Sweden have taken advantage of their national registries to begin asking questions about the PANDAS hypothesis, as they say right here, association of strep throat with mental disorders, testing key aspects of the PANDAS hypothesis in a nationwide study. And this was done by a group in Denmark using their registry that contains information from basically first mom's appointment for a prenatal exam up until the child's current age for 1,067,743 children. All of them less than 18 and about half girls and half boys, as you would expect. Of those more than 1 million children, 638,000 had received a strep test during the study period, and 349,982, almost 350,000, had positive test results at least once. So this is a beautifully nested case control study where they were able to just go into this data registry, look at the children who had had a documented strep infection and children who had not had any documented strep infections, and compare those two populations. And they had beautiful results showing that any kind of streptococcal infection had increased the rate of any mental disorder. Now, we had been talking about OCD and tics, and yesterday in the question, some other symptoms came up, psychosis, ADHD-like behaviors. My personal fear, manic depressive behaviors, because of the emotional ability, and if the child had a strong family history of bipolar disorder in one or both of the parents, then I think the clinicians would perhaps rightly assume that that child's presentation of extreme emotional ability, suicidal depression might be a symptom of that. But in fact, this evidence would suggest that even that kind of presentation was increased, the risk of that was increased after strep infection. It was much more highly increased for obsessive compulsive disorder, as had been originally hypothesized, and much to the neurologist's dismay for tic disorders as well. Furthermore, the risk, they were able to compare the risk of strep versus the risk of other infections, because that had been another criticism, right? Not only is strep frequent, but kids are getting sick all the time. And what they found was that although the results weren't as strong, probably because the numbers get smaller at this point in time, that any strep infection had also increased the risk of mental disorders, OCD, and tic disorders above that incurred by having had a non-strep infection. So this is lovely evidence of a direct connection. It's partnered by a study that was done in adult patients with obsessive compulsive disorder, suggesting that they actually had had increased risk of strep infections as children. So nice sort of big epidemiologic data. Caution with any epidemiologic study, it can only tell you an association. We talk about increased risk. That's actually not an appropriate term. It just means that they were more likely to have that diagnosis if they had that. You can't make any connection. And if you want to think about this, then the sort of how the statistics work, the very strongest correlation and the strongest risk of drowning actually comes from eating ice cream, because there's a very high correlation between ice cream consumption and drownings. Well, you guys have common sense, and you know that that's because both of those go up markedly in the summertime. But if you're just looking with a tiny little microscope, you might not make that association, and you'd end up saying eating ice cream makes you drown. So with that caveat, this study is even more exciting than the Danish study, because this is an interactive, prospective, longitudinal, population-based study done by the European Multi-Center Tic in Children's Studies, the MTICS group, which is comprised of investigators from 16 European institutions. This was a paper that was published earlier this year, talking about the methodology of this study, which is huge. It's been going on for a long time now, and they're actually getting ready to release some of the results. And because of Dr. Graziella Orefici, who's sort of there in the middle, Dr. Orefici was able to, from the very beginning, get strep as a potential risk factor for the development or worsening of tics within this cohort of patients. And we're excited to see what they're going to be able to find in this study of more than 700 children. So we'll be watching for that one probably later this year, maybe by 2020. And Kristen and I were talking last night that 2020 is the year that we are all going to be able to celebrate many, many wonderful things. So here's to the future. Well, the second piece, and as Dr. Pasternak said yesterday, this is really a seminal study. It's truly a life-changing, quite definitive work, and we had hinted about it yesterday. I think I even showed you this slide. I just want to make sure that you understand how important this is, because in rheumatic heart disease, they still have not successfully created an animal model in which they can provoke rheumatic carditis in a mouse or a rat model. And yet Dr. Agaliu, Dr. Cleary, and their colleagues were able to invoke strep infections in that nasal-associated lymphoid tissue and produce changes in T cells that trafficked into the brain and actual demonstration of neuroinflammation as a result. So if people say, PANDAS can't exist because strep doesn't do X, Y, and Z, this is the study you point to, Dileepan, D-I-L-E-E-P-A-N, and it's there at the bottom in the journal Clinical Investigation. This one was from 2016. I think some of you heard from Dr. Cutforth when he was in BC that they're continuing this work and actually looking not only at the role of strep, but now beginning to look at the role of the CSF changes in our affected children. The key here was the multiple infections, and it fits beautifully with data that Tanya Murphy had, and we talked about a bit yesterday. In that Putnam County, Florida study, one of the most striking things was that the children who had a single strep were more likely to have tics, choreiform movements, problematic behaviors, but children that had at least 2 or 3 infections during the school year were the most likely. And so that would fit with this model. It also answers that question of why Joey gets it in this classroom and all of his classmates had strep but didn't end up having PANDAS. Well, maybe Joey, this is the 3rd or 4th time he's been exposed within a relatively short period of time, and he has activated T cells. That extrapolation is probably just as dangerous as trying to make a causal relationship out of an epidemiologic study, because that's a huge jump from a laboratory-induced process in a mouse to what's happening in a child in the classroom. But it's nice that the clinical data and the basic science data are at least in concert with each other. So the question of molecular mimicry is the one that we went after first. I wish I could find the little piece of notebook paper that Dr. Henrietta Leonard and I were scribbling on one day when we were trying to think about all of these pieces in this model. We had been studying together rheumatic fever, trying to understand everything that we could at the time, and had laid out this cartoon back then and sort of were asking questions next to it. And we kind of circled antibody-mediated symptoms because it seemed like the easiest one to go after because of a study done by Husby in 1976, in which he took serum from patients with Sydenham chorea or with lupus and compared them against how the serum reacted against strep. And what he found out, and then he went to the brain. So that was a beautiful experiment in which he actually showed that the antibodies in the Sydenham chorea patients recognized not only the brain tissue, but also the strep bacteria, whereas the lupus patients only reacted against the brain. So there was something cross-reactive in those antibodies. It also was a wonderful place to sort of draw our circle of interest, because in our simplistic model, it meant that plasmapheresis was going to be the very best treatment for Sydenham chorea and for PANDAS, because what it basically is a blood cleaning procedure. We're going to talk about it more this afternoon, but in plasmapheresis, after 5 sequential treatments within a week to 10-day period, you have removed 95% of the circulating antibodies, and because of equilibration across the system, you've decreased the antibody load for the host as a whole by nearly 50%. So it's just go in, scoop them out. And again, in our simplistic model, this was just a misdirected immune response. And we thought, get rid of those aberrant antibodies, get rid of the problem children, and everybody will be fine. Well, it turned out it wasn't quite that easy. There's clearly more pieces to that. And we now, after 25 years, know that those antibodies are present. They are actually bioactive, and I'll show you in a moment the most recent class of antibodies that's been recognized in PANDAS children, but it's probably not sufficient, and there's a lot more going on. And one of the other reasons that plasmapheresis is so effective is it's a reboot of the immune system. It's not only pulling off 95% of those circulating antibodies, but it's also pulling off complement and elements in the cascade. It's impacting on the T cells, and it literally is sort of a nuclear bomb when what you'd really like to be using is a very fine chisel. So this is just a composite slide to show you the work that Dr. Madeline Cunningham and her colleagues, Christine Curvin and others, have done at the University of Oklahoma over the last few years. And the development that I had hoped would be in press by now, and is not, is that Dr. Cunningham did these same experiments on the serum and the CSF from the children participating in the Yale NIMH IVIG trial. And what we found out was that the Cunningham panel actually does work relatively well. It is higher during acute illness than it is during convalescence. It was disappointing that there wasn't a particular pattern that predicted either symptom severity or the sort of likelihood to respond. However, the combination of a Cunningham panel being positive in at least 2 of the antibodies and the CAM kinase, plus a positive anti-nuclear antibody, an ANA test, was predictive of response to IVIG. So in terms, I told you that I have a conflict now. I will potentially be getting royalties off the Cunningham panel. But for the clinicians, if I were going to order a Cunningham panel, I would only do it if I was really trying to decide, is this child in an immunoreactive state in which IVIG might be useful? And then I would only do it in combination with the ANA. And we didn't look, but my suspicion is that if you had a very high ANA, you might be just as likely to have a response to treatment, and you wouldn't have to bother with the Cunningham panel. So hopefully that sounded less biased than someone who has royalties might. This was a study published by Frick, F-R-I-C-K, Luciana Frick, and Chris Pittenger was the senior author out of Yale University, again using serum from our children who were participating in that IVIG trial. And the nice thing about that trial was because there were half of the kids got placebo and yet had a second spinal tap at that point, we can look at children who improved. 2 of our kids improved during placebo, just probably from antibiotic response, and see the kind of changes without intervention. What they found was very high titers of anti-cholinergic acetylcholinergic interneurons. That's the red in the middle. And when they put the overlay, you see that it gives you orange. In the control subjects, there was actually, this is mislabeled. It's backwards. I apologize. The control should be on the right and the PANDAS on the left, because that is the positive one there on the bottom where it's merged, and you see there's orange double staining. It was very specific in that they did not find it against other cells within the striatum, within the stroma of the basal ganglia, and they have extended this now to show differences between the acute and the convalescent serum, as well as the presence of these antibodies in CSF samples. So I don't know that this is going to turn out to be our equivalent of the anti-NMDA, but I think it's actually more likely to be useful than the specific dopamine D1 and D2 receptors, lysoganglioside, or the tubulin that was chosen for the Cunningham panel, because those actually came out of Dr. Cunningham's research on rheumatic heart disease and the dopamine from experiments that her colleague in Israel had done, and all of those were in vitro rather than an in vivo application. So if you summarize that finding, what they found was that there on the bottom, cholinergic interneurons are independently implicated in OCD and tic disorders by postmortem studies and targeted ablation in mice. Dr. Pittenger's group had actually found a family in Ecuador with a very high prevalence of Tourette syndrome, and what they found was that this family had had a genetic abnormality of this particular cell population, and that was how they had initially made the link. Elevated binding was quite specific. In Dr. Pittenger's study, they did not find increased binding to D1 or D2 expressing motor striatum neurons or to cholinergic neurons in the medial septum. So it was a very specific finding, and he's doing a lot of work right now. In fact, has just been given a NIH grant to further work on this hypothesis and to track the work further. So lots and lots of questions remain. And to try to begin to answer those clinical questions, I just used the model that had been used in autism. And when I took a decade break from PANDAS and went to work in extramural, my job was to sort of wrangle all of the autism research and advocacy groups. Was quite a challenge, but what we found was that in autism, unlike PANDAS and the parents of PANDAS patients, they didn't work well together, and we had to incentivize them to do a consortium-wide study. And unfortunately, we work too well together, so they are not incentivizing us to do this. And my wonderful colleagues have been basically volunteering to give us this information for a very small investment by the NIH. Relatively small investment, I should say. What the NIH has done is to provide the database, clinical trials database, and a clinical trials secure input where parents can actually get their own unique identifier, enter all of the data, and then the clinician can go in, pull it up for their clinic visit, but it's also being stored along with samples that are required from that patient. The investment of about $600,000 a year maintains our ability to get samples into the sample repository, have them processed so that in the future any investigator can access them. And this is what I was saying, we need to fill this repository with 1,000 well-characterized children as quickly as possible. Unlike some of the other registries that have been started, this one is going to be controlled because the secret to, in my opinion, just my opinion, the secret to genetic research is a well-defined phenotype, and that's where autism failed. In the autism community, autism is a syndrome like PANS, but there's a failure to acknowledge that. And they act like when you have a diagnosis of autism or even autism spectrum disorder, that child A is the same as child B, despite the fact that they always say, if you've seen one child with autism, you've seen one child with autism, right? And in that consortium, they actually had, because the psychologists were putting the data in, the difference between a child being in the affected group and the unaffected group was a score on their ADAS and their ADIR, where literally 1 point make difference on a 40-point scale makes that diagnostic cutoff. Clinical judgment is used at the NIMH in our studies, and we spent almost as much time characterizing children with autism as we had to do to determine whether a child had PANDAS, because it isn't just a score on the ADAS and the ADIR. It goes further than that. That's a lot of negative talk to just say, I know that some of the parent groups in the United States have been frustrated that I haven't just opened this up and let them fill it for us right away, because they could. There are hundreds, if not thousands, of children in the US who have a diagnosis of PANDAS, but if I could be blunt, garbage in is garbage out. So if we aren't very careful about who gets into the study, then we may not be able to find something that was truly there. Consequently, we've established common measures, and we just had a meeting at the NIH in February, where we had representatives from 18 different institutions who all came together. We had been hammering out these common measures by email and teleconference for years going into this, and we still haven't gotten to the point where every site is using the same forms, because I don't think it's necessary. What we have is that every form being used in the 18 different sites contains the same basic information, and with the data dictionaries we'll be able to cross those platforms and come together. They're asked to confirm the diagnosis and put in enough information that an independent validator can go back through and validate those diagnoses, as well as entering severity ratings, so that we have some ability to determine what factors are influencing this. And then we have protocols for collection and storage of the biologic samples. That was actually quite easy because the NIMH had contracted with Rutgers University to create the NIMH biorepository for genetic samples, and they were willing to extend that to include serum and some of the things that we need to hunt for antibodies and other etiologic factors. So the common measures, the ability to get the data in, get it back out, and the sample repository were what NIMH was creating, and then until I left, we were serving as the site for the sort of continuing checks on diagnostic validity, cross-site reliability, and training, et cetera. This effort now needs to be picked up by an extramural group, and I'm hopeful that our colleagues at Columbia may be able to do it since they have currently a grant studying obsessive compulsive disorder and maybe could get a supplement to add this in at least until we can get a planning grant together to get the work done. One thing that did seem to work and was able to help Stanford get their program going in their early days, as well as Arizona, Minnesota, Florida, Columbia, and others, was that while I was still in intramural, we were able to provide a research assistant to each of those programs for a year's period of time to get those data. Dr. Mike Cooperstock has now retired at the University of Missouri, and we were able to actually provide a research assistant to redact all of his clinical charts and clinical information, as well as to recode his samples, so those will also be coming into the repository. My hope would be that we can add a couple of the Canadian centers here into this. Dr. Purnell had come to a meeting that we had to talk about the diagnostic work and sort of when we first launched this notion of the centers, and Dr. Rosenberg has been an ally from the very beginning, so hopefully Calgary, Saskatoon will get all of the major players in Canada together helping us with this. And if you want to have your own bank, as long as the two of them talk to each other, that's fine with me too. One of the key reasons that we need to do this is that as we get these samples and data in, we obviously need to have people ready to ask the questions and meaningfully get the answers together. And so the NIH is being petitioned by the U.S. Advocacy groups to fund PANDAS PANS research to a greater degree. Our current institute director, I guess I would just characterize him as an agnostic, and so it's going to take some work to convince him it's helpful, but at least he is not as anti-clinical research as the previous director was, so I think we'll be in good shape. And that was all I had. Thank you.
Thank you very much for that update. Okay.